but then the introns are removed, okay, right? The 3' end, there's a polyadenylation happens. Basically, multiple adenine units are added, and so that modification takes place. So what you see is, you know what, if this is your DNA, that's going to be your RNA, right? At least this is true for prokaryotes. For eukaryotes, if this is your DNA, probably this is your RNA. You see the difference? The number of nucleotides has changed because there is splicing. Sometimes, you know, maybe at the tail there's a few more adenine nucleotides added, poly-A tail. It could be as big as, I don't know, 50 adenine nucleotides, or maybe even 500, sometimes even 1,000 adenine nucleotides are added. So when you have something going from DNA to RNA in the same size, it's referred to as collinear. It's referred to as collinear. When you don't have that, it's referred to as non-collinear. Mentioned here, non-collinear. So many large RNA molecules observed in the nucleus were absent from the cytoplasm, suggesting that nuclear RNA undergoes some kind of changes before it exits from the nucleus and goes into the cytoplasm. This is true in case of eukaryotes like us, like human beings like us, right? So gene organisms, so that's linear and collinear. Now this is a slide showing you the same example. Look at this. This is a DNA molecule. You see that it undergoes transcription, and you have a messenger RNA form, and then you have proteins. So all this looks like the same size. Now, obviously, they started drawing these circles bigger because every 3 codons code for 1 amino acid. So obviously, you're going to have 3 times less. But once again, it's collinear. But in eukaryotes, you don't really see this collinearity because of modifications that happen. Now, how did they really realize that non-collinearity is actually seen in eukaryotes? So what some of the people working in the field of research scientists did was, if an RNA is synthesized from a DNA, right? For example, if this is your DNA and this is your RNA, right? So these strands should unwind, and then a new RNA strand is synthesized, right? This RNA strand, if it is synthesized from this one, obviously it should be complementary to this strand. So there's complementarity between these 2 strands, right? So what they said was, let's actually try to hybridize the DNA and the RNA, even though the only difference is uracil basis. So more or less it's kind of, to a significant extent, these 2 strands should be complementary to one another. So they took DNA here, as shown here. They also took the RNA, and they kind of heated up. You know, I said, like, when you do PCR in the lab, you heat up the DNA and then the DNA unwinds, right? And then eventually when you cool it down, DNA rewinds back again. Double strand DNA actually reforms. So they added DNA and RNA and they heated it up. So when this DNA kind of unwinds, they were expecting that one of the RNA strands should at least go and bind to the DNA and form a DNA-RNA hybrid. In principle, it should, because the sequences are complementary to one another. But then what happened was, they found that most of the cases, DNA kind of, one of the strand of DNA kind of, it base pairs with the second strand, not really with the RNA, because they realized that there was, even when there was a complementarity between DNA and RNA as shown here, you can see this image here. It shows there is this loop-like structure indicating that there is something that's not complementary between DNA and RNA. So that was the first key, or that was the first indication which said that probably at least in eukaryotes, there is not really complementarity between DNA and RNA. So the RNA that is synthesized is kind of changing or doing some modifications. So that brings us to introns and exons, something we have heard me say in the previous lecture. So introns, I like intervening, I for introns. So intervening sequences are called introns. They do not code for any protein, usually. Well, there is now some new evidence showing that introns can also kind of, depending on how it's spliced, intron sequences can also be included in proteins. But, let's not go into that. But for the sake of the chapter that we're studying, let's understand that introns are intervening sequences. They intervene, basically they have nothing to do. In other words, introns can be considered as some kind of junk DNA, okay? Many eukaryotic genes contain coding regions called coding and non-coding regions. The coding regions are referred to as exons, and the non-coding regions are referred to as introns. Non-coding intron, coding exon, okay? Introns are usually seen very commonly in eukaryotes like us, but in prokaryotes, you hardly see them. I mean, there are some rare exceptions, but for the sake of this chapter, let's say that introns are only found in eukaryotes and not in prokaryotes. Okay? Now, with respect to introns, so these introns which are part of the messenger RNA, they have to be deleted, they have to be kind of, well, the right word is not deletion, the right word is spliced, basically cut out. If the introns have to be cut out, how are they cut out and what are the different classifications of introns that we have? Now, introns can be divided into 4 different groups. For example, group 1 introns, you find them sometimes in some bacteriophages. Like I said, introns are usually found only in eukaryotes, very rarely in prokaryotes. So just remember that introns are only in eukaryotes, not in prokaryotes. But here I just quote some examples saying that you can also find them in bacteriophages and some eukaryotes. So group 1 introns, these RNA regions have the capability to splice themselves out. They have their self-spliceable capability. So that region, if you take the RNA, if the RNA has these introns have the capability to cut themselves out of the RNA. That is group 1 introns. Group 1 introns, they can self-splice. Basically, they can cut themselves out. Now, group 2 introns, they are found in mitochondrial genes and chloroplasts. Group 2 introns. These group 2 introns also have the capability to splice themselves out of the messenger RNA, but the way that they are spliced out is different from how the group 1 introns are spliced out. Understand? So both group 1 and group 2 introns, they can basically splice themselves out from the messenger RNA. Group 1 introns are found in eukaryotes and some bacteriophages, but group 2 introns are found in mitochondria and in chloroplasts, mitochondria and chloroplasts. Now the third group of introns are actually the nuclear pre-mRNA introns. So this is the most common form of introns that we see. Normally when we discuss about eukaryotic transcription, we say that in humans, or for example, we always say, we take the example of yeast, which is a fungi, and fungi is actually a eukaryote. So you find these nuclear pre-mRNA actually is the most commonly discussed intron. So basically, if you say mRNA, that messenger RNA has introns and exons. Let's say these mRNA are coding for some proteins. They have introns, and these are the most commonly studied. Now, these introns that we see, for example, let's say in human genes, the splicing or the removal of these introns requires small nuclear RNAs. Small nuclear. So basically, we studied small nuclear RNAs, small nucleolar RNAs. Then we studied microRNAs. So we studied different classifications of RNAs in the previous chapter. Remember? So some of these small RNAs, a specialized class of RNAs called small nuclear RNAs, they kind of help in splicing out the introns that are found in the pre-mRNA. Okay? Also, to remove the introns in the pre-mRNA, like human genes or in yeast, there's something called spliceosome. We'll come to that in the upcoming slides. Spliceosomes are actually protein machineries that can bind this mRNA. So if you have an mRNA like that, So this mRNA has, so it has an intron here. So there is a protein complex that actually binds to it and cuts it out. So in addition to this spliceosome, which like I said will be coming up in the next slides, which actually helps in removing these introns, the small nuclear RNAs are also involved in splicing. Is that clear, guys? Now, how are these small nuclear, I know the small nuclear RNAs, microRNAs, you know, this small piece of RNAs, how are they actually generated? So let's say if you have a RNA, right? This is how an RNA looks like. Let me just take you to the previous slide, it's easier to show you that. Guys, if you look at this slide, okay, this is the primary structure of an RNA, right? The one shown in green. It's like a single strand. Sometimes the RNA has the ability to fold on itself, okay? When it folds like this, it forms something called secondary structures. Now, whenever proteins in the cell, there are some proteins like Agnaut or dicet. These proteins, when they see a double-strand RNA, by the way, this is not a double-strand RNA, it just happened to fold on itself. When it sees, what they do is, these enzymes will, they will cleave here and they will cleave somewhere here. That makes small pieces of RNA, get that? So these small pieces of RNA are actually generated from a messenger RNA. If you take a long messenger RNA, somewhere in the tip it happens to fold on itself and enzymes cut it out. When they cut it out, you have like 20, 30, 40, 50, 80 nucleotides of RNA falling out. These small pieces of RNA are referred to as microRNAs or small nuclear or small nucleolar RNAs. And they actually have a very significant role in gene regulation. For example, right now I said here that small nuclear RNAs are involved in splicing the messenger RNA. So splicing mechanism is also a way of gene regulation, because the RNA is being cut out into pieces and stuck together, so you are regulating the protein expression at that level. So that's why I say the small nuclear RNAs can process the pre-nuclear RNA. Now the fourth kind of introns are actually found in transfer RNA. You remember I said the transfer RNA, one of the main roles of transfer RNA is that it binds to one amino acid and it transports this amino acid to the ribosome so that the ribosome can link this amino acid to the growing peptide chain. So these transfer RNAs have the fourth classification of introns, right? And the removal of these introns in the transfer RNA requires some kind of special enzymes. Is that clear, guys? So basically, in other words, I spoke about linearity and collinearity between the DNA and RNA, and I said in prokaryotes it's collinear, but in eukaryotes like us it's not collinear. The first thing. Second, I spoke about introns and exons, and then in introns I said there are 4 different classifications of introns. The first classification of introns, they can splice themselves out. You see them in some bacteriophages and some in eukaryotes. The second group of introns is found in mitochondria and in chloroplasts. These introns can also cut themselves out. They can splice themselves out, but the splicing mechanism here is different from group 1. And group 3 introns are actually found in the regular messenger RNAs that we usually discuss. And the removal of these introns requires small nuclear RNAs and also requires something called spliceosome, which I said will come in the upcoming slides. And the fourth group of introns are found in transfer RNA. And these removal of these introns requires some special enzymes. Now, is actually 2 different genes to show you ovalbumin gene and the cytochrome B gene. In these 2 genes, what you see here, this dark olive green is actually your exon. These are the regions that actually code for the protein. In between, you have this light olive green, which is your intron, and this has to be removed. For example, in ovalbumin, you have a total of 8 exons. In cytochrome B gene, we have a total of 5 exons. Now, after splicing has happened, these introns are actually removed, and then you have a total of 8 exons placed one next to the other, and this is actually your mature RNA. Now, this will be translated into protein in eukaryotes. So these introns are found almost always in, or let's say they're found in eukaryotes and not in prokaryotes. Okay? So this albumin has 8 exons and this cytochrome B actually has 5 exons. They're just showing you 2 different genes, telling you that the number of exons between different genes is actually different, the number of exons. Okay? Now, right, different types of introns. You can look at the stable column, group 1, 2, nuclear tRNA, pre-RNA, tRNA. So these 4, there are 4 different groups of introns and how they are spliced out. See, this is, here it's spliceosome also does it, and also small nuclear RNAs can splice it out. And here it's different enzymes. Now looking at the structure of the messenger RNA itself. In bacteria, mRNA is transcribed directly from DNA, but in eukaryotes, what happens is this mRNA is first synthesized as pre-mRNA. Now the so-called splicing happens, a poly A tail is added, so basically a lot of adenine nucleotides are added at the 3' end, and then it's moved into the cytoplasm where translation happens. Okay? Each amino acid of a protein is specified by a set of 3 nucleotides called the codon. So if you look at an RNA, every 3 nucleotides is grouped as one codon. Okay? And that 3 nucleotides in the RNA will code for one amino acid. Both prokaryotic and eukaryotic messenger RNAs contain 3 primary regions. Okay? It could be bacterial or eukaryotes, 3 regions. The first is the 5'. So basically, the 5' region you have, in the end you have the 3' region, in between is actually the coding region. So the 5' region, basically the beginning of the messenger RNA is referred to as the 5' untranslated region, or also called the leader sequence. Because ribosomes do bind on the 5' region. So if you take the whole RNA, at the very first step you have this 5' region. The ribosome binds there, it slides over, but then it does not synthesize any amino acids from that, from the 5' region. When it slides a little bit further down, there it sees the first codon, which happens usually almost is actually methionine, which is coded by the codon AUG, and from there actually translation begins from there. So the 5' region is referred to the 5' untranslated region, also sometimes called as the leader region. The leader region of the 5' untranslated region. Now second, and then you have the protein coding sequence, which is the middle region, and then at the very tip we have the 3' untranslated region called the trailer region. Okay. Now, for example, in bacteria, when you're looking at this messenger RNA, this bacterial messenger RNA has a Shine-Dalgarno sequence. I think we discussed about this sequence in the previous lecture. Shine-Dalgarno sequence is basically nothing but consensus sequences. I said consensus sequences are nothing but conserved nucleotides. You find these sequences in more than one bacterial species, for example. So that repeat of that nucleotides in that order is consensus. Now the use of a consensus sequence, okay, the consensus Shine-Dalgarno sequence. For example, the use of the sequence is when you have that nucleotides like UAA, GGA, GGU, something like that. The ribosome can nicely bind that sequence. The folding of the ribosome is kind of very comfortably fits exactly on that sequence. Even if you happen to change one of those nucleotides or 2 of those nucleotides, the ribosome cannot sit properly on that. So that's the significance of the sequence is that's those nucleotides in exactly that same order can be bound by the ribosomal proteins very easily. Now this consensus sequence, it's called consensus sequence because it's kind of conserved from species to species. For example, when you look at the DNA, when you go back in the previous chapter I said when you look at the DNA sequence, the DNA has a promoter region, right? This promoter region has the starter box and some other consensus sequences. So these sequences are conserved from species, and transcription factors, which are some proteins and RNA polymerases, can bind those consensus sequences. Like that, in bacteria, the Shine-Dalgarno sequence, which is shown here as UA, GGA, GGU, you don't have to remember the sequence, but I'm just saying, this sequence is actually recognized by the ribosome, and the ribosome binds on it. Once the ribosome binds on it, it begins to slide forward, and by the time you have the first initial 5' region ends, you have the first initiation codon is AUG. AUG codes for methionine. Once the ribosome sees the AUG, then transfer RNAs, bring in the proper amino acids, and the ribosome kind of begins to form the peptide bonds between different amino acids, creating a protein. So the second region is called the protein coding sequence, and the third is the 3' untranslated region, also called as the trailer region. So 5' untranslated region, which is called the leader sequence. In the very end, you have the 3' untranslated region, which is the trailer region. In between, you have the protein coding region. Okay? The 3' is also known to affect the stability of the RNA. Well, for that reason, even the 5' is also known to affect the stability of the RNA. And then later, since this, what is not eukaryote, so I'll not speak on it. Okay. So look at this. This is the full length of your messenger RNA. This is the 5' untranslated region. So you have the Shine-Dalgarno. So if you look at this RNA, I would say this RNA is actually a bacterial RNA, because I see this Shine-Dalgarno sequence here. Right? The ribosome binds here, and this is the start codon. It goes on. You have the stop codon. You have the 3 different stop codons. So either of the stop codons is found here, and obviously termination happens. Protein synthesis is terminated by the time the ribosome slides up to this region. And below that, you still have the untranslated region. So basically, the messenger RNA extends beyond the stop codon, the messenger RNA. But the protein synthesis does not extend beyond the stop codon. Is that clear? Okay? Now, looking at the 5' leader sequence, I said 5' untranslated region. This 5' region is known to increase the stability, because the 5' region, there is actually a guanine nucleotide added. The guanine nucleotide is added, and then to this guanine, a methyl group, CH3 group, is added. It is said that when the 5' region is linked to a guanine nucleotide, and if you add a methyl group to it, it actually protects the messenger RNA from being eaten up by proteins within the cytoplasm. There are proteins which actually are involved in degrading messenger RNAs, right? If you have a messenger RNA, ribosomes bind and protein is synthesized. After the protein synthesis happens, you don't need this messenger RNA anymore. It needs to be removed from the cell. So for removal of the messenger RNA, there are proteins that degrade the messenger RNA after the message has been translated to protein. Now, once you have the 5', where the 5' region has this cap, that actually protects the messenger RNA from abruptly being degraded by some proteins. Okay? So, and this guanine nucleotide probably is not found in the DNA, but it suddenly shows up in the RNA. Because, once again, like in eukaryotes, I said there's a lot of editing that happens to the messenger RNA. One of the editing is adding like 3 to 500 poly in A adenine nucleotides at the very 3' end. The other modification is adding a guanine nucleotide at the very beginning. And the third modification is to this guanine nucleotide, a methyl group is further added. So basically, whatever gene that is, like I said, like in prokaryotes and eukaryotes, so what you see in eukaryotes what you see in the eukaryotic gene doesn't really translate into the messenger RNA, because a lot of modifications happen. And one of the modification is the addition of this guanine nucleotide, and this guanine nucleotide is linked to a methyl group that is known to protect the messenger RNA. So this kind of 5' cap and 3' cap, 3' poly A tails are seen in eukaryotes, not in prokaryotes. Is that clear, guys? The poly A tail is seen in eukaryotes like us, not in bacteria, not in virus. Now, Look at this here. This is a messenger RNA, right? Do you see this messenger RNA at the where the 3' end? You have, for example, some nucleotide. This could be an AGCT, what AGCU, any one of them, right here. You also have a second nucleotide here. But if you look carefully where I'm pointing with the mouse, you don't see a guanine here. Do you see a guanine here? No. Is that big enough for you to see? All right. So you don't see a guanine nucleotide here. But if you go further down, do you see a guanine here? So a guanine is suddenly added by some protein, some enzymes, whatever they do. So now this guanine nucleotide, it's only guanine. Now do you see a methyl group added to the guanine here? So that addition of the methyl group, and also once the methyl group is added, for the next 1 or 2 nucleotides, again further methyl groups are added, CH3 groups. When these methyl groups are added to the guanine here and also to the second and third nucleotide here, this is considered the 5' cap region. It provides some kind of security, some protection for the messenger RNA. So the cytoplasmic proteins don't eat it up. Somehow they recognize this guanine with the methyl groups and they kind of leave it alone, at least until it's translated to protein. Okay? See, most eukaryotic mRNAs have a 5' cap. This is the 5' cap region. It's magnified here. See, this is the guanine here, this is the methyl group, and this is the ribose sugar. Okay? Now, also in eukaryotes, what you see is the addition of the poly A tail. So, by the way, if you see, let's say, 300 or 400 adenine nucleotides added to the 3' end, you should not assume that probably they were 200 or 300 thymine bases in the DNA. You understand what I'm trying to say? So you will not find 200 to 300 or 500 thymine nucleotides in the DNA, but you will find 200 to 300 or even more adenine nucleotides added to the 3' region of a messenger RNA. This is once again a modification. So these nucleotides are not encoded in the DNA, so you don't find them. So if you have an A, you're supposed to have a T in the DNA. You don't have those T's, but still you have this A's. That's why it's referred to as polyadenylation, multiple A's. You could have as little as 50 to 70 adenine. You could have as much as, I don't know, 500, even 800 adenine nucleotides added to 3' region. See the poly A tail addition of 50 to 250 nucleotides, and also this poly A tail facilitates attachment of the ribosome in the mRNA and plays a role in export of mRNA to the cytoplasm. So transport of the messenger RNA in eukaryotes is kind of somehow favorable. Addition of the poly A tail favors the proper transport of the mRNA from the nucleus into the cytoplasm. So you can see this. This is your DNA, and this is after transcription you have the RNA. By the way, guys, even though this is your RNA, there are some multiple A adenine nucleotides within your RNA. This is not the 3' end. So this poly A happens here. So you look at this here. This is the poly A. There are some repeats of adenine also within the messenger RNA, and these repeats of A within the messenger RNA actually help in splicing of the RNA. I'll come to that in a minute. But don't confuse between these few adenine nucleotides here and this poly A tail here. Okay? The poly A tail is at the very tip of it. Now, so this is the DNA. You have the messenger RNA synthesized from it. Now it's cleaved. So basically, there are some cleavage sites. Now after the cleavage sites happens, then now this becomes a new 3' region, and then adenine nucleotides are added. Now this messenger RNA is ready to be transported into the cytoplasm. Look at this table column. It just tells you about the addition of the poly 5' cap, 3' cleavage, and poly A, RNA splicing, and we'll come to RNA editing in a minute. RNA splicing and editing is something we have not covered yet. We'll come to that in a minute. But now let's look at RNA splicing. So now, till now we spoke about adding a guanine nucleotide to the 5' region. 3' region is linked with a lot of adenine nucleotides. Now how does how does the protein machinery within the cell recognize which is the exon and which is the intron, right? If the protein machinery is going to splice the RNA, it can cut anywhere, right? And also probably some in proteins, some of the amino acids could be lost and the protein could eventually become non-functional. So how does the splicing machinery referred to as a spliceosome? Look at this here, spliceosome. How does the spliceosome know where to cut and how? We'll come to that in a minute. So one of the functions of the spliceosome is actually to recognize this few repeats of adenine nucleotides within the messenger RNA here, shown here in purple. I'll come to that as to how they recognize and what they do. So now these spliceosomes, which is machinery that cut the RNA, they also recognize some small and short consensus sequence located within the messenger RNA itself. I'm not speaking about the Shine-Dalgarno sequence at the 5' end in bacterial messenger RNA, but you have other consensus sequence located between before and after every intron within the messenger RNA. Now first, so constant sequences and the spliceosome. Splicing requires the presence of 3 sequences in the intron. One is, the first sequence is, one of the end of the intron is referred to as the 5' splice site, and the other one is the 3' splice site. Okay, so if you look at look at this. Guys, if you look at this messenger RNA, right, here is exon 1 shown in dark green, and this is intron shown in light green. You have the second exon here, okay? Exon, intron, exon, that's what we see. If you look at this here, this is the 5' site and this is the 3' site. If you happen to cut here, let's say if you cut this piece here out, automatically this tip becomes your new 5' site. Wherever you cut, it becomes a 5' site. Now, if this is the exon and this is the intron, so which is actually a full sequence, there are some sequences shown here like A, G, G, U. So the end of an exon usually ends with AG, and the beginning of the intron usually starts with GU. Now these 4 nucleotides, along with a few more nucleotides here, this whole complex is also considered as a consensus sequence by the spliceosome. You get that? So in fact, if you, for example, here you see they say C by A. So you can have either a C or an A here, and then you have AG. So let's say this is CAG, that actually codes for one amino acid. Even though that codes for amino acid, those three nucleotides here are also considered as part of the consensus sequence by the spliceosome machinery. And they sit on this sequence and they cut it. So at the other end of the intron and the beginning of the exon 2, you have CAG, and then the exon begins with G. So these 4 nucleotides in the RNA are also recognized as the 3'. By the way, this is the 3' end of the RNA, I get it. But imagine there's a cut here, and now this region, this part is now, this after the G here, this is considered again as a 3' region because there's a cut. So those nucleotides are once again recognized as a consensus sequence. Now you have to understand that the nucleotides are not only used for translating protein, but some nucleotides in the exon, some nucleotides in the intron together can be recognized as a consensus sequence, and then that's how the spliceosome or the protein machinery can recognize these sequences and cut it out. So the consensus sequences recognized by the spliceosome are, the first one is, one at the end of the intron is referred to the 5' splice site, and the other at the end of the 3' is 3' splice site. These splice sites possess short consensus sequences. These are the short consensus sequences which I showed you a minute ago. Now, second, most introns in pre-mRNA begin with GU and end with AG. So GU and AG. So this GU here is the starting, the AG here is the ending. Okay? So most introns have this GU and AG at the beginning and the end. And the third is that there are a few adenine nucleotides within the mRNA itself which are also recognized. Which are these few mRNA nucleotides? I'm referring to these ones here. These are also recognized by the spliceosome machine, okay? Not these at the very end. Now, sorry for jumping back and forth because I just want to show you the images to make things easier for you to understand. Now, splicing takes place within the large cluster, large structure called the spliceosome. This spliceosome actually has, 5 RNA molecules inside it, and it also has short proteins, almost up to 300 proteins. So basically, you're looking at a huge complex of proteins that actually sit on the RNA and they cut it at specific sites. So multiple protein units or protein subunits coordinate to bring about the splicing of the RNA and then joining it together. Now look at this. So this is the splicing of the pre-RNA, exon 1, exon 2, and this is the intron. And this a few adenine nucleotides here are referred to as the branch point here. They refer to the branch point. Now looking at this is exon 1, this is exon 2, now this is the intron. What happens is, the spliceosome machinery, the protein machinery sits on it, they cut here, shown with the red arrow here, and they kind of fold it in such a way and they stick it here. Now where are they sticking? They're sticking this piece on the intron itself. Now what is this region? This is the region where I was pointing to the few adenine nucleotides. Remember that? So those few adenine nucleotides are here. So the spliceosome cuts it, folds it, and sticks on it, sticks the intron on itself, and then pulls it out. That pulled out intron is referred to as the lariat. And now this lariat, which is nothing but the intron, is actually cut into very small pieces and completely destroyed. Now you have exon 1 and exon 2, and the intron in between has been removed, and then exon 1 and 2 are actually joined together. So now this is your mature RNA. This is a clean message. Basically now this will be transferred into protein. They only showed two exons here for our understanding, but assume that there are multiple exons here. Okay? Now, looking at this a little bit in detail with the spliceosome. So look at this once again. The same steps I'm repeating, but here I'm showing you some of the subunits of the spliceosome machinery. So this is exon 1, exon 2, and these red arrows are the cut sites. Now you see U1, U2, you see here U4, U5, U6. So all these are subunits of the spliceosome machinery, Now these subunits, for example, U1 binds to the junction between exon 1 and intron 1. U2 actually binds in the branching A tail region here in the center. Now U4, U5, U6, they bind to the exon. They kind of fold it in such a way that one end of the intron sticks on the branch point A region, and that region is removed as a lariat, and then exon 1 and 2 are joined to one another. Is that clear, guys? Yes, any questions on this? Any doubts on that? So that is actually how usually splicing happens. So those small constant sequence at the ends of the introns and exons, spliceosome machinery sits on it, it cuts it, it folds it and sticks it at the intron on itself and then pulls it out. Now you also have some small splicing mechanisms. Some introns in the pre-mRNA are removed by special processes called minor splicings. These introns have different consonant sequence at the 5' splice site. And even here the spliceosome machinery is involved, but these are somehow referred to as the minor splicing events. Now, self-splicing introns, I think we have looked at group 1 and group 2 introns here. Earlier I said have the ability to splice themselves out. So these introns, they fold in some way that they kind of splice themselves out from the messenger RNA. They do not require this spliceosomes to remove them out. Now, when you're looking at splicing, there are alternative process for splicing. One is called the alternative splicing pathway. The other one is called the multiple 3' cleavage site. What is the alternative splicing pathway? There is more than one way to splice a messenger RNA, because one way to splice a messenger RNA is by removing the exons. Another way to splice an RNA is to remove the exons and sometimes also remove some of the exons also. So this is called alternative splicing. Splicing can be achieved in more than one way to yield different messenger RNAs that are translated to different proteins, because if you remove the exons also sometimes, obviously the proteins get shorter and the amino acids in this protein are different from the first one, basically a few less. So the folding of this protein could be different and that protein could entirely have a different function. So this is called alternative splicing. I'll show you the image. And there is something called multiple 3' cleavage sites. You remember I showed that the beginning of an exon can also have some consensus sequences. Sometimes in the exon itself, in the middle also you have this consensus sequences, and sometimes splicing happens inside an exon. So when an exon is cut inside it, that is referred to as the multiple 3' cleavage sites. In these cases, 2 or more potential sites for cleavage and polyethylation are present in the pre-mRNA. Both alternative splicing and multiple 3' cleavage sites can exist in the same pre-RNA transcript. So let's look at it. Look at this. This is alternative splicing, okay? The normal way that you know of is, this is exon 1, exon 2, and exon 3. So the introns here are removed, and then eventually you have, if you look at this here, exon 1, exon 2, and exon 3. This is your messenger RNA with the poly A tail added, okay? This is how we know. Now for the alternative splicing mechanism, if you look at this here, splicing has happened in such a way that exon 2 is also removed here. That's why in the alternative splicing mechanism here, you have exon 1 and exon 3, and exon 2 is actually removed with the intron here. You see that? So this is referred to as alternative splicing. Sometimes one or more exons can also be removed. That is alternative splicing. And the other one which I referred is to the multiple 3' cleavage sites. For this one, what you see is, see, this is exon 2, right? Within the exon 2, there will be some consensus sequences, and sometimes the spliceosome machinery, what it does is, it cuts within the exon itself. So here, if you look at it, this messenger RNA has, this is exon 1 shown in green, this one in yellow is exon 2, but the same messenger RNA is spliced within the, spliced here within the exon. Here it is not spliced within the exon, that's why the exon 2 here is longer compared to exon 1 here, you get that? So within the exon itself there are splicing sites. When those spliced sites are used to cut, exon 2 is shorter. That's how you see it here. Exon 2 here shown in yellow is shorter, whereas exon 2 here is longer because the splice sites happen to cut at the very end of the exon 2. Do you get that, guys? Guys, is it confusing? One second, we need a break, 10 minutes, I'll give you 2 breaks, if that's what you want. You're not responding, side. Am I going too fast? Hey, say something, it's okay. I see absolute silence, I didn't get it, so. Break or short, I'll give 2 breaks. You want the break? Can take it? No, yes? Guys, come on, say something. You can talk.
Okay.
You know, let's take a 7-minute break and then I'll give another 7 minutes.